parte del presidente dell'Accademia, professor Massimo Vincenzini, che non può essere presente, ma che mi ha incaricato di darvi il benvenuto. È chiaro che l'argomento che affrontate è guardato dall'Accademia con la massima attenzione, perché è la massima attenzione che la comunità scientifica e la pubblica opinione dà a queste problematiche, cioè alla difesa. La difesa con metodi diversi da quelli dell'impiego dei prodotti della chimica di sintesi. Ecco, cioè questo è un grosso argomento sul quale tra l'altro l'Europa ha richiamato più volte l'attenzione e qualche volta ha anche fatto delle imposizioni, non sempre per l'ambiente mediterraneo perfette, però sono regole che l'Europa dà. Per cui benvenuto, benvenuto davvero ogni sforzo che viene da una, direi da una parte della comunità scientifica molto responsabile. Io conosco Paolo Pesaresi da molto tempo e so l'impegno nella attività scientifica e anche il valore. Quindi davvero con molto piacere accogliamo questo convegno finale, mi sembra di capire, no? Convegno finale di progetto, che peraltro le strane coincidenze delle volte c'è, come si chiama, l'eterogenesi dei fini. Domani a Firenze, non qui, ma all'Auditorium Cosimo Ridolfi, che quindi porta il nome di un formidabile georgofilo dei primi dell'Ottocento. Cosimo, in quell'auditorio faremo un convegno che riguarda questo stesso tema, cioè nuovi metodi di lotta. E c'è anche un punto interrogativo nel titolo, quindi è finita la chimica di sintesi? Punto interrogativo, eh? Punto interrogativo. Però sarà molto interessante seguire quell'intera giornata al mattino con relazioni selezionate e poi nel pomeriggio una tavola rotonda che prevedo dalle domande che sono state fatte, ma soprattutto dalle risposte che sono arrivate, prevedo che sarà una, perlomeno diciamo un brainstorm. Ecco, non andremo molto più là in quella sede forse del brainstorm, ma l'intenzione vera di dare magari successivamente, non in quell'occasione, ma di dare un'informazione alla pubblica opinione, questo sì, è molto attesa. Quindi davvero grazie a voi per questo e poi il discorso continuerà domani in altra sede. Prego Paolo.
Allora, grazie innanzitutto all'Accademia dei Gergofili, al professor Massimo Vincenzini, al professor Amedeo Alfi e al professor Piero Craveli che ci hanno permesso di fare questo convegno finale del progetto NoPEST che tra pochi minuti vi illustreremo in questa prestigiosissima accademia, in questa bellissima sede e ovviamente in questa meravigliosa città. Siamo veramente onorati di poter presentare i risultati del nostro progetto qui all'Accademia dei Gergofili, qui a Firenze, e speriamo che le persone qui in aula e anche le persone che sono collegate online li possano trovare di loro interesse. Questo convegno ha di fatto l'obiettivo di poter mostrare alla comunità scientifica, ma anche a tutti quelli che noi generalmente chiamiamo gli stakeholders, quindi alle aziende, agli stessi operatori del settore degli agrofarmaci, ha l'obiettivo di mostrare i risultati che abbiamo ottenuto in 6 mesi scusate, in 6 anni di sperimentazione. Mi sposto adesso dall'altra parte perché ho una piccola presentazione che introduce il progetto. Abbiamo appunto come l'ambizione di mostrarvi i risultati del progetto NoPEST, Novel Pesticides for a Sustainable Agriculture, finanziato dalla Commissione europea nell'ambito del programma Future Emerging Technologies. È un progetto che è iniziato nel gennaio del 2019, è stato inizialmente finanziato per 5 anni. Questi anni poi sono diventati 6 perché ci siamo trovati in mezzo al periodo dell'emergenza Covid e quindi abbiamo richiesto un'estensione di un altro anno. E come vi dicevo, oggi cercheremo di mostrarvi i risultati di questi 6 anni di sperimentazione. Il progetto NoPEST coinvolge 5 paesi, 4 paesi della Commissione europea, come potete vedere Italia, Svezia, Francia e Spagna, e un paese associato, Israele. E più nello specifico, il progetto ha coinvolto 6 partner, tra cui il Royal Institute of Technology di Svezia, l'Università Bar Ilan in Israele, l'università che nel 2019 aveva il nome di Paris Sud, che poi è diventata Paris-Saclay, francese, l'Università di La Rioja in Spagna e il partner industriale, per l'appunto Sipcamoxon. Questi partner ovviamente sono fatti di tante persone e in questa immagine siamo noi a Milano al kick-off meeting e poi in questi anni si sono uniti molti ricercatori, devo dire molti giovani ricercatori, quindi oltre all'attività sperimentale, NoPEST è anche servita a formare molti ricercatori, alcuni di questi sono ancora oggi con noi che portano avanti le attività sperimentali di NoPEST. Altri hanno ricevuto un buon training nell'ambito di questo progetto e questo ha consentito loro di trasferirsi in altri laboratori sia in Europa che negli Stati Uniti. Il progetto NoPEST nasce di fatto nel 2019 in un contesto come quello del European Green Deal, dove da un lato l'Europa, in particolare nella strategia Farm to Fork, chiede di poter ridurre del 50% l'utilizzo dei pesticidi convenzionali entro il 2030. E dall'altro invece c'è la necessità degli agricoltori di avere a disposizione pesticidi perché sono l'unico modo per poter garantire la produttività agricola così come la conosciamo oggi. In realtà questa necessità aumenta di anno in anno del 2,7%. Quindi di fatto da un lato la riduzione, dall'altro la necessità di pesticidi, quindi l'urgente necessità di fatto di trovare delle alternative ai pesticidi convenzionali. Ecco, all'epoca con la professoressa Simona Masiero abbiamo iniziato a ragionare se potevamo fare qualcosa per cercare di trovare una soluzione a questa esigenza. E guardando nella letteratura ci eravamo accorti che c'era un grande interesse da parte della comunità scientifica internazionale nell'utilizzo di piccoli peptidi, quindi di sequenze amminoacidiche fatte di pochi amminoacidi da utilizzare nell'ambito della protezione delle piante. Come potete vedere dal numero di pubblicazioni, tra il 2012 e il 2022 c'è stato un aumento quasi esponenziale di pubblicazioni su questa tematica. E le ragioni sono piuttosto semplici. Il meccanismo con cui funzionano questi piccoli peptidi è piuttosto intuitivo. Di fatto si tratta di avere delle piccole molecole di pochi amminoacidi che interagiscono fisicamente con degli enzimi bersaglio, con delle proteine bersaglio prese, isolate, identificate dai patogeni e a seguito di questa interazione fisica si spera che l'enzima, la proteina del patogeno possa essere bloccata e di conseguenza possa venir meno la capacità del patogeno di infettare la pianta o addirittura di uccidere il patogeno. È sicuramente una tecnologia sostenibile da un punto di vista ambientale, perché utilizza amminoacidi naturali, quindi questi peptidi, una volta rilasciati nell'ambiente, verranno poi degradati in amminoacidi naturali. Ed è un meccanismo anche molto specifico, ha la stessa specificità di quella che potremmo immaginare di una chiave all'interno di una serratura. Quindi c'erano numerose ragioni per cui la comunità scientifica si sta concentrando su questa tecnologia. A quel punto si trattava di capire su quali patogeni sviluppare questa idea. E qui è stato molto utile il dialogo che abbiamo avuto con il dottor Pietro Quersola, responsabile della ricerca e sviluppo di Sipcamoxon, perché con lui e in collaborazione con la professoressa Silvia Toffolatti abbiamo capito quali erano le reali necessità in ambito industriale, le necessità che avevano gli agricoltori. E ci siamo in particolare abbiamo deciso di concentrarsi in particolare su Phytophthora infestans, che è uno dei principali patogeni della patata, e su Plasmopora viticola, che è un patogeno molto importante per la vite. Ecco, identificata la tecnologia, identificati i nostri target, abbiamo allora incominciato insieme alla professoressa Masiero a mettere in piedi la strategia. E quello a cui abbiamo pensato è, innanzitutto, occorreva individuare i target di questa tecnologia, cioè quelle proteine, quegli enzimi verso i quali indirizzare i nostri peptidi. E qui abbiamo identificato uno dei gruppi leader in Europa per quanto riguarda l'analisi della parete cellulare degli omiceti, e in particolare il gruppo del professor Vincent Bulone e di Vaibhav, che lavorava all'epoca nel laboratorio del professor Vincent Bulone, che in questi anni poi ha messo in piedi il proprio laboratorio. Simona Masiero, che è la persona che ha immaginato per prima la possibilità di utilizzare questi peptidi per poterli utilizzare contro patogeni in agricoltura, è la persona che ha messo in piedi la tecnologia per poter in tempi rapidi identificare i peptidi che potessero interagire fisicamente contro queste proteine bersaglio. Abbiamo poi chiesto alla professoressa Tolfolatti, sempre dell'Università di Milano, di aiutarci a mettere assieme dei saggi in vitro o ex vivo per poter testare l'attività antimicrobica di questi peptidi, e in collaborazione con la professoressa Sala Pellegrino siamo riusciti anche a mettere in piedi una strategia per poterli sintetizzare chimicamente in maniera rapida. Successivamente la professoressa Sandrine Ongeri dell'Università Paris-Saclay e il professor Anok Zenderovitz della Bar Ilan University in Israele hanno contribuito permettendoci di rendere questi peptidi molto più stabili nelle condizioni ambientali e di aumentarne l'affinità per i bersagli. E inoltre il professor Anok Zenderovitz, utilizzando questi peptidi, ci ha aiutato anche a identificare altri tipi di molecole che noi abbiamo chiamato non-peptide small molecule, possibilmente di origine naturale, che potessero avere anche loro un'attività antimicrobica. Infine, parallelamente all'identificazione di peptidi, il professor Javier Tordaguglia dell'Università della Rioja in Spagna ha sviluppato tutta una serie di sensori nell'ambito dell'agricoltura di precisione che oggi consentono di identificare in una fase molto precoce la presenza del patogeno e quindi di poter spruzzare queste molecole solo in quelle zone in cui l'infezione sta avvenendo. E infine, tutta questa tecnologia, grazie alla collaborazione del partner industriale Sipcamoxon, è stato possibile verificarne la funzionalità in campo, in condizioni reali di campo. Ecco, gli obiettivi di questo progetto quindi erano identificare peptidi, sia lineari e ciclici, che avessero un ruolo contro gli omiceti e in particolare contro Phytophthora infestans e Plasmopara viticola, migliorarne l'efficienza attraverso lo sviluppo di peptidomimetici, di cui a breve sentirete parlare. Utilizzare degli approcci chemoinformatici per identificare molecole non peptidiche, possibilmente di origine naturale, che avessero anch'esse un'attività antimicrobica, e quindi disegnare e migliorare dei sensori per poter essere utilizzati nell'ambito dell'agricoltura di precisione. Ecco, queste sono di fatto le tematiche che adesso i nostri speaker affronteranno nelle prossime 2 ore e vi mostreranno i risultati che hanno ottenuto in questi 6 anni di sperimentazione. Gli interventi che abbiamo previsto sono mediamente di circa 15 minuti. Al termine di questi 15 minuti possiamo avere almeno 5 minuti di discussione sia con le persone che sono qui in aula, ma anche con le persone collegate online. Chi è collegato online semplicemente alza la mano e poi gli verrà data la parola. Quindi io concluso questa parte, lascio la parola alla professoressa Silvia Toffolati e da questo momento gli interventi saranno in lingua inglese.
Buongiorno a tutti, spero che mi vediate e mi sentiate anche bene. La presentazione sarà in inglese, adesso la condivido con voi. Intanto ringrazio moltissimo Paolo per l'introduzione e per avermi dato questa opportunità di raccontarvi quello che è stato il nostro ruolo come patologi vegetali all'interno di questo progetto veramente emozionante anche per certi punti di vista. Allora, presentazione. Ok. So, I'm talking to you about the results that we achieved in screening for efficacy, the peptide aptamers that were selected by the group of Professor Simona Masiero and collaborators. Of course, when you have to test the efficacy of the products, you find challenges. And now I will describe you how we tried to overcome the difficulties in screening for fungicide efficacy when you are dealing with different pathogens, which are oomycetes and have different lifestyles. So what we want to address with this talk is to establish a path to an innovative plant protection through the fungicide efficacy screenings. So you all know, also from the introduction of my colleagues before, that biotic threats are reducing our yields worldwide. Each year we lose approximately 20 to 40% of our crops due to biotic threats. Imagine that you have also a biotic threat and also other disasters that are occurring each year. The problem is that these biotic threats reduce not only the yield, so the amount of food that we can provide, but also the quality and the nutritional value of the food. So they hamper our food security in a very damaging way. Each year, our the agriculture world is challenged by several problems. We know that plant protection products, so the products that we apply in agriculture to protect the plants from, in this case, biotic threats, are necessary for achieving a good yield. The problem is that our farmers are facing a reduction here in Europe, particularly, of the products that are available for protecting the plants due to the regulatory issues and the safety of these products for human health and the environment. So we have each year less and less products. And on the other side, we also have parasites developing resistance to these products. So it's every year it's more challenging planning a disease management strategy that is able to let us produce enough for an increasing population. But NoPEST has some solutions to that. So as Professor Pesaresi told us before, luckily we have these substances, the peptides, aptamers, which can help us in dealing with the problems that I just highlighted. So here is a cartoon where you can see, imagine a fungal spore that is germinating and producing a germ tube, and it's starting to imagine infecting a plant tissue, of course. The peptides that we are developing in NoPEST are targeting oomycetes, and they are targeting in particular the cell wall biosynthesis. If a spore a fungal spore is attacked by imagine the peptide aptamers, it will not be able to produce, to biosynthesize the cell wall. And so it will not be able to produce a germ tube, and so it will not be able to infect the plant. So in this cartoon, we are happy because actually the pathogen is not able to infect the plant. And this is good. So we know that we are designing these peptides to be effective against oomycetes. But how do we assess their efficacy? We can assess it in different tissues and in different environments. We can start with laboratory tests where we assess the inhibition by these peptides on the pathogen structure development or on the ability of the pathogen to infect on plant tissues, such as leaves or parts of leaves. We can assess their efficacy in greenhouse. We try to assess if these peptides that are selected by laboratory trials are able to protect also the entire plant. So we move from the laboratory to the greenhouse and we do in planta tests. So we test the efficacy on living plants. And in the end, the selected peptides that were able to be effective in laboratory and in greenhouse are assessed for their efficacy in natural condition. So we go from control condition, laboratory and greenhouse, to natural conditions, where the pathogen inoculum is not controlled and the environmental conditions are not controlled. They are natural. So we see the performance of our peptides also in a natural condition. But first, we start from the in vitro test. Starting from the in vitro test that we perform in laboratory, we have to choose which kind of in vitro test we have to perform. And in this case, I'm showing you a type of test, a very common test that everyone can perform, also in a not well-equipped laboratory, which is assessing the growth of the pathogen on a growing when it is growing on a medium. Here is on an agarized medium. Here you can see an inoculated petri dish where the pathogen hyphae are developing in the medium. And in this case, what you are doing is assessing the inhibition by the peptide or your antifungal agent of mycelial growth, and you can also assess sporulation if you then collect the spores produced by the pathogen. And we are able to perform this type of assay when we can grow the pathogen in axenic culture, so with cultivable parasites, but not all the parasites that we are dealing with are able to be cultivated. We also have, we are dealing also with uncultivable parasites, obligate parasites of the plant, which are so well adapted to their host plants that we are not able to cultivate them in axenic cultures, so on a liquid medium or agarized medium. For these parasites, we need to assess the efficacy of the peptide on living tissues of the plant. In this case, we are using leaf disc assays. So, as I told you, we have different pathogens, so we have to develop different tests. Here you can see the symptoms of the late blight disease on the potato tubers, and also on the leaves. This disease is caused by Phytophthora infestans, which is a very damaging biotic agent. Maybe you know all of the tragedy that occurred at the middle of the 19th century in Ireland, when millions of Irish people died due to the loss of their yield due to the presence of Phytophthora infestans. It was a tragedy occurring in Ireland, and we still are facing the threat by Phytophthora infestans. Luckily, we have many more means to control this pathogen nowadays, but as I told you, we have a lot of problems also with this. Phytophthora infestans is a pathogen which is cultivable on artificial media, for instance, pea agar, which is very easily prepared, is a suitable medium for growing Phytophthora infestans. And for the NoPEST project, we use a CBS culture, a CBS isolate, which was isolated from tomato. For Phytophthora capsici, which is another damaging agent, it is causing blights on cucurbits, and here you can see some symptoms on all the parts of the plant. We also were able to test the efficacy of the peptides on the growing culture of the pathogen, and we used a CBS strain isolated from pepper. Pythium is a genus of different encompassing different species which are causing very damaging disease, as damping off on seedlings and root rot and seed rot. Here you can see root rot on sorghum, and these agents are very, very important. For the Pythium category genus, we tested the efficacy of the peptides against Pythium ultimum varietas ultimum, a CBS strains also in this case, which is cultivable on artificial media. So the preliminary tests that we performed for these three species were performed on the inhibition of the mycelial growth, while for Plasmopara in actinic culture, while for Plasmopara viticola, which is the last oomycete that we tested, we are doing a different kind of test because Plasmopara viticola is an obligate parasite of grapevine and we cannot cultivate it in actinic culture. Here you can see some symptoms of the disease. Plasmopara viticola is able to affect all the green parts of the plant, the leaves, the shoots, and the inflorescences, and also young berries. And we cannot isolate it and cultivate it in pure culture. So we used for the NoPEST project our strain collection of the Department of Agricultural and Environmental Sciences. We had in our project, we had a deliverable on the setup of in vitro assays to assess the efficacy of the peptides. Here is the outline of our deliverable. So for the in vitro assays, we developed protocols which are freely available for the assays on synthetic media, on leaves, and also we also prepared a media appendix where everyone can find the media that are needed to perform these assays. When we assess the efficacy of the peptide, we have to think, what do we want to check? Usually, we check first the pre-inoculation with the pre-inoculation assays, the preventative activity of an active ingredient. So first, we assess if the molecule is able to inhibit the growth of the pathogen when it is applied before the pathogen inoculation. Then we also performed post-inoculation assays. So we tried to assess if the peptides also have a curative effect. So if they are able to block the growth of the pathogen when it is already growing. Usually when we develop our disease management strategy in field, we develop the strategy by preventing the infections. But it is also true that sometimes the infections are already occurring, and if we have a product that it is able to block the infection, it is a very good thing for our farmers. In each test, we developed different tests on solid medium, on liquid medium, in 24 and 96 wells. And in each test, of course, we have to assess the growth or infection of the pathogen on negative controls, sterile distilled water and water solvent, if we had the necessity to use the solvent, but our peptides were selected to be soluble in water. And then you always need positive controls, and we have a fungicide, which is affecting the cellulose synthase A3, mandipropamide. And also we had another peptide, which is NoPV1, which was developed in a previous project, and it was effective, so we use this as a positive control as well. So once you decide which is your in vitro test, then you start doing it. So you always have to think that you have your negative control, your positive control, and then what do you do? You assess usually first you do a discriminatory dose assay. For the peptides, we decided to screen their activity at a very high concentration, which was 100 milligram per liter. And the peptides that were able to inhibit the pathogen growth at that concentration were further tested. And in this further test, we did a dose response analysis where we exposed the pathogen structures to increasing concentrations of the peptide to find which was the the lowest concentration, the lowest efficacy concentration. And we did this in agarized medium, in liquid medium, and on leaf discs with Plasmopara reticola. The molecules that were able to develop to show an efficacy in these in vitro assays were also tested in implant assays in greenhouse, where we did pre-inoculation tests. So we treated the plants, then we inoculated them, and after an incubation in humid chamber, we scored the plants for the symptoms. So in this case, you just look at the symptoms or the sporulation, which is occurring on the tissues. We did these implant assays with Plasmopara reticola, as you can see in this slide, but also on tomato for the phytophthora infestans. In the end, what do you do with all the data that you collect? Of course, you have to find the way to express them, to discuss them also with the company which is going to develop the product. So which is the best index that can be used is the protection index. So you express your data in a number, a percentage, that is expressing how good performed your peptide in terms of protection. And you can do it for on agarized media, and especially it's more suitable for leaf disc and implant assays. For the post-inoculation efficacy, I'm showing you this type of assays that we developed, that Demetrio Marciano, the PhD student working on this project, developed. He inoculated sporangia or mycelial pieces, because you have to imagine that not all the oomycete are able to produce sporangia easily, so sometimes you have to use mycelium. So he also had to develop a way to standardize the mycelial inoculum. And then he inoculated this liquid medium treated with the peptides, and he measured absorbance at time 0 and also at 2 or 3 days after inoculation. And then he was able, based on the absorbance, to calculate the percentage growth inhibition, which is another type of information that we can use for selecting the molecules. Then we had to decide which were the most effective peptides, and we decided that they should have a protection index not significantly different from the positive control, which is the fungicide or the other peptide, and they should provide an efficacy higher than 80% at the discriminatory concentration. So this was our workflow, the way that we used for checking our efficacy of our peptides. we assessed the efficacy of more than 54 peptides on different pathogen species. 24 were linear and 30 were cyclic, and 30 were cyclic, sorry for the error. And from these, we selected 2 peptides, which were then formulated by the company, Camoxon, and then assessed in field finally. In July, we had the opportunity to visit one of the experimental fields. These 2 peptides were assessed both on tomato and on grapevine. And in this case, the tests are quite different. They follow the EPO standards with randomized blocks, 4 replicates per treatment, 5 plants per replicate, and also, in this case, negative and positive control. In this case, the positive control was copper because the company thought that the best thing is to use these peptides also in organic farming. And I cannot say to you that much, but the peptides performed quite well. Stay tuned for more information. So in conclusion, for my part, it's always important to get knowledge about our pathogen to select the best method for testing. The method should be high throughput, and one of the problems that we have to face with peptides is that they are highly costly when they are synthesized, chemically synthesized. So this means that you have a very low concentration, low amount of peptides, and you have to test many different species. So you should need a lot of peptides. So we have to find the most suitable way to assess the efficacy of these peptides on different species with very low amount. So that's why we use the liquid media assay, which was very, very useful for us. It is useful not only because the peptides are costly, but also because when you do this type of assay, you produce a lot of garbage. And this is going to be a cost for the environment. So the more high throughput is your test, the better it will be also for the environment, because you have less products to dispose of. Then our laboratory and greenhouse tests were fundamental to select the peptides with the best peptides, which actually showed a good efficacy also in field trials. And field trials for us are really important because with field trials, finally, we assess the efficacy of the peptides, not only on some tissues or organs of the plant, and typically we assess on leaves of the plant, but also you can assess the efficacy on the fruits, which is really important, and also in uncontrolled conditions. That's all from my side. I'm grateful, and I wish to thank Demetrio Marcianò, which did a lot of work during his PhD on in vitro assay, and he did a very good job. I'm very happy and proud of him. And also Giuliana Maddalena, which is there with you. So she can also, you can also contact her if you want to ask questions, because Giuliana developed the implant assays that I just showed you. I'm really sorry I cannot be there with you, but I have a health problem, and so I'm here in Milan. But I'm, of course, staying here if you have any questions. Thank you for your attention.
Thank you. Thank you, Silvia, for this very nice presentation. As Silvia said, that this was one of our very first task to develop antimicrobial assays in order to test the properties of the peptides that we have identified. So we have a few minutes for some questions. I don't know whether there are people online or here from the audience that wish to ask questions to Silvia. Okay. Okay, so if there is no questions, then I think we can, thanks again, Silvia, for the very nice presentation.
Thank you.
And now, Now we can move to another important aspect of the NoPEST project, that was studies conducted at the level of the home-isolated cell wall with the aim to identify candidate protein, candidate enzymes that should have been the target of our peptides. So they should have been the protein to inhibit by the physical interaction of our peptide. And that has been actually the main task of the laboratory that is conducted by Vaibhav Sevastani. And Vaibhav is now going to report about the analysis that his lab, together with his collaborator, have conducted on the cell wall of home isolates.
Okay, good afternoon, everyone. It's lovely to be, in this historical center. My name is Vaibhav, and I work at KTH Royal Institute of Technology in Stockholm, Sweden. Okay, so maybe I should go back. So if you see, the title of my talk today is Targeting the Cell Wall, a Novel Approach to Disease Control. So this is one of the term, I think maybe many of you may not be familiar with that, what is a cell wall. So without going in detail about the NoPEST project, I think let's check, that what the cell wall is. Most of the cell types are surrounded by a cell wall, whether it's a bacteria, it could be fungi, it can be plant cell. It has many functions. It provides basically cells with a structural support, shape, protection, and it also functions as a selective barrier. And if we look at the structure of cell wall, structure and composition of cell wall in different organisms, for instance, on this slide, you can see bacteria, plants, fungi, and algae. Basically, what is common between, among all these organisms is the cell wall is made up of complex carbohydrates, maybe carbohydrate, the term which we are familiar with. And it's also in general, it is called as glycans and some proteins. Theoretically, there can be, numerous combination possible for the complex carbohydrate structure. But in nature, there has been very selected in terms of specificity and function for this cell wall in different microorganisms. So what can be concluded from this slide is cell wall is different in different microorganisms. And I think that's basically, the take-home message from here. And you will see, that how exactly it has been used in the context of NoPEST. So when we talk about NoPEST project, we are, as you already know, Paolo has already said, Sylvia has also mentioned it, we are working on a group of microorganisms which are classified under this family called oomycetes or water molds. And they are, as you can see, here on this picture, that they have a separate family. So they look like fungi, but they are not really, connected with fungi, for instance. And these group of microorganisms, they are pathogenic to both animal and plants. And as a result, they cause heavy losses to both aquaculture and agriculture industry worldwide. Some of the examples, for instance, you can see here on the right-hand side of this slide, for instance, Saprolegnia parasitica, which is also oomycete pathogen. It's a fish pathogen actually affecting fish and aquatic animals. Then you have already heard about Phytophthora capsicii, Phytophthora infestans, which are affecting different plants, for instance, tomato, potato, eggplants. Then we also have Phytophthora ramorum, Pythium ultimum. One of the important one in the context of NoPEST is Plasmoptera viticola, which is affecting grape wine. So in NoPEST, mainly we are working on plant pathogenic oomycete species. Okay. So if we look at the structure of oomycete, so here on this slide, there is an example. You can see a potato leaf is infected by this plant pathogenic oomycete, Phytophthora infestans. This is a recent study which came in Yemen. This is the first report of existence of Phytophthora infestans there. So they are basically everywhere. So these are thread-like, fungi-like organisms, and they also, you can see thread-like structure here, and which also contains cell wall. And they reproduce by basically something, a specialized structure, which is called as zoospores, and which are also lately responsible for the infection process, for instance, in this case, potato or other crop plants. So what is basically the global impact of all these plant pathogens? I think Sylvia has already covered very nicely, in the previous talk. And she also mentioned about this, which is an infamous example, this Irish potato famine in mid-1800, which is basically associated with the oomycete pathogen Phytophthora infestans, which basically killed all the potato plants. As a result, there was a starvation and death of almost 1 million people. And then it also caused a heavy immigration, from that region. And in this picture, you can see a mother and her son. They are looking for a healthy potato. And this picture is actually from that time. So if you look at the FOA estimation, FOA stands for Food and Agricultural Organization. They say that every year up to 40% of crops are lost due to plant pathogens. And what does correspond to it? Basically more than $200 billion losses worldwide. So of course, these numbers are definitely, quite significant. And maybe these are a little bit old figures, so definitely it has increased. So now we talked about, of course, plants, diseases. So what can be the disease control strategy? For instance, so traditionally what we have been using is the use of fungicide, which basically represent the largest, almost 46% of pesticide sales in the EU market. If you see on the graph on the right-hand side, the countries which are basically the major consumers, they are France, Spain, Italy, Germany, and Poland. Interestingly, these countries are also the largest producer of grapes, potatoes, tomatoes, and all these different crops are highly susceptible to oomycete pathogen. Which pathogens are these? For instance, Plasmopara viticola, Phytophthora infestans. Both these pathogens, we are working on this NoPEST project. So disease control has always been a priority, both in healthcare sector or even agriculture. But now the traditional approaches, for instance, the use of antibiotics and pesticides, they have serious issues now. And what are these issues? One of the major problem is the increased resistance of plant pathogen towards fungicide. So basically, the fungicide won't be active, or it will basically not work. And what other impact we have, it is a negative impact of these pesticides on microbial diversity, because it is also killing basically what are the healthy microbes. As a result of this decreased microbial diversity, it also impacts the plant health and carbon dioxide soil retention. So then, now if you remember from the beginning, then why this cell wall can be a promising target. And so there must be, some advantages. So here, we have tried to put some advantages, what cell wall targeting would bring as compared to chemical treatment, for instance. So when we talk about the resistant developments, there is a lower risk. The reason is that pathogen will basically, will have hard time modifying its cell wall. And so there is a lower risk for antimicrobial resistant development as compared to chemical treatment. Then we also have this target essentiality, which means that cell wall is a very important target. And if you break the cell wall, for instance, in case of pathogens, it will basically die. So it's a very essential target as compared to, the different signaling pathways or metabolic pathways which are targeted through chemical targeting. It is also a pathogen specific, because as I mentioned that, cell wall are different in different organisms. So basically, if you are targeting pathogen cell wall, definitely it should not affect plant cell, for instance. And it should also be basically safe for humans, even though human cell doesn't have cell wall. Then it also has most of them are biodegradable agents, so the environmental impact is very low. It also has, the immune system synergy. So what exactly happens that when there is a break of the pathogen cells, the material inside the cell which comes out, which basically enhances the plant immune response as compared to chemical treatment, for instance. So it also has a long-lasting effect, which also means that you don't need to overuse it, and that can be comparable with the biological control and integrated pest management system, for instance. So as you have already seen, the NoPEST, one of the main NoPEST goal is to develop a new generation of innovative and environmentally safe pesticides. In our case, is basically peptide aptamers. And then, of course, once we have it, it's the treatment of plant pathogens or oomycetes in vine or crop production supported by precision farming. So what was our role, from KTH was basically to understand the cell wall polysaccharide structure. And when I say cell wall polysaccharide, basically it means carbohydrates, and biosynthesis. And when we say biosynthesis, so there are proteins or basically enzymes which are responsible for the synthesis of these different carbohydrates. And then, of course, to validate the efficacy of these peptide aptamers basically in the lab condition. Here, I just want to show you an example about the cell wall biosynthesis in fungi. I mean, here you can see that the fungal cell wall on the top and side here, these are different carbohydrates which are present in fungal cell wall. For instance, one of the carbohydrates is chitin, and then, of course, there are several glucans. And you can also see that there are several enzymes present, on the cell wall or even inside the pathogen, which are responsible for the formation of these different complex carbohydrates. So basically, our role, as I mentioned earlier, in this project was also to do something similar in case of oomycetes pathogen. So what we did, we started actually, digging in the genome of all the different oomycetes pathogens, the one which we were working on, and, came up with a list of candidate cell wall target enzymes. And there are many enzymes, which are listed here on this side, right-hand side. And so what we also did, since we identified these different cell wall enzymes, since we didn't know that exactly, that what's their function, we also did something called biochemical and functional characterization of selected cell wall enzymes from selected microorganisms. And then, that was basically, for the protein part or enzyme part. In addition, we have also done cell wall characterization, which is basically for carbohydrates, because we didn't know which type of carbohydrates are present in the cell wall. So here, what you can see, these are basically, something called simple sugars or monosaccharide in a more scientific word. Here you can see a comparison of different sugars present in 3 different species, oomycetes species. And just to show you, an example, for instance, in the middle, you can see glucose. And so glucose, can also be present in different types of polymer, as you can see here, like which glucan stands for glucose. And here, they are present in a different way. They are attached to different carbohydrate polymers. So we have basically, that information from different oomycetes pathogen. What we did, we basically compiled all the information which we got it basically, from the identification of protein enzymes which are related to cell wall in different pathogens, and also the different carbohydrates which are present in different pathogens. And then we made a comprehensive map of the cell wall metabolism in these selected oomycetes, where you can see, I don't need to go in detail, of course, it's a complex figure. But you can see, we have identified basically, most of the proteins and carbohydrates which are crucial for the pathogen. So just to conclude, in this project, we have identified many cell wall related enzymes. We have also done biochemical characterization of some of these enzymes. In addition, we have identified different cell wall carbohydrate polymer. Altogether, we have made a comprehensive map of cell wall metabolism for selected oomycete species. And we have also done identification and testing of peptide aptamers against these targets. But Simona will talk more about, in the next presentation. So altogether, if we combine the results from the NoPEST, it has the potential to reduce dependence on traditional antibiotics or pesticides, in case of crop protection. And here is a model which we have little bit proposed, based on the information we got about the oomycete cell wall, where you can see different different enzymes which we have identified, and the different cell wall carbohydrate polymer from the oomycete pathogen. I would like to acknowledge, of course, the whole NoPEST consortium and also the funding from the EU. And here, all the pictures you see on this slide, these are basically different members from the KTH team who has been working, during all these years. And with this, I would like to thank you all again. And if you have any questions or comments, I'm happy to answer. Thank you very much.
Thank you, Vaibhav, for this very nice presentation. So as you have seen, Vaibhav has given a major contribution to identify the targets that we try to inhibit by the use of our peptides. I don't know whether there are some questions from Vaibhav, either here or online. No? Okay, then thank you again, Vaibhav, for your contribution to the NoPEST. And so, as we said, we have now the assay that we can use to test the activity of the peptides. Thanks to the work of Vaibhav, at this point, we knew which are the proteins that we could target using the peptides. And now, obviously, we need a technology that allow us to identify in a fast and cheap way, I would say, those peptides that can interact physically with the target protein and eventually inhibit the activity of those target proteins. And the technology was indeed developed by Professor Simona Masiero at University of Milano. And Simona is going now to show us the technology that is at the basis of the NoPEST project.
Okay. Thank you, everybody, for listening what we have developed in the lab. And when the NoPEST project started, actually the group that I represent, most of this group has been now spread all over the world somehow, felt like the drum of the Brunelleschi dome, because we had the duty to feed most of the work package with the molecules. So we needed to feed Silvia and the group of the pathologists to test something. We had the pressure to feed the French group for modification and so on. So it was actually a difficult pressure. And how can I go? So what we have developed in the frame of the NoPEST project was a technique suitable for isolating antimicrobial peptides. Actually, antimicrobial peptides, it's a bit maybe confusing word. That's why in the project we use a aptamer word. So what are peptides? Peptides are, in the case of antimicrobial one, are defensive molecules. And actually, all living organisms going from fungi to mussel, to insect, but also human being, produce them to defending themselves. And their mode of action is quite specific. So, and they are nowadays used in several fields in biomedicine. They are used as new antibiotics because they are able maybe to lysate the membrane of the pathogen. They are used in the regulation of intracellular target, and in this case, they might be used to control even cancer and other important pathologies, and they are also very well used for to control the immune system. So for human problem related to alteration in the immune system, this type of molecule are perfect. So our idea, it was to use this molecule in agriculture and to use them not to target pathogen for human being or for animal, but to target plant pathogen, which means to have a further problem because it means to use this molecule in very large field. So you need to cover thousands of hectares of lands that are cultivated. But we didn't like the idea to look for something that is a very broad mechanism of action. We rather prefer to work with drug discovery, but targeting drug discovery. So these three guys have been acknowledged with the Nobel Prize, and these are the so-called poor biomedicine Nobel Prize. That's how I like this definition, because they all focused in one human disease. Campbell and O'Moore, they were, for instance, interested in filariasis, while Yu too, she was interested in malaria. And basically their idea was to look for antimicrobial peptide, testing thousands of molecules that come from nature and looking for one that works. It's like to look for an eye in a high pack. So it's a very difficult work and very long and tedious. They have been inspired by the work of Fleming, the one that discovered between the two world wars the penicillium. And this discovery was again a bit very, how to say, a matter of luck, because Fleming had a plate of E. coli that was contaminated by penicillium. He went home, so like any of us, he was fed up by the wrong work in the lab, throw away the plate, but then coming back home, realized on something that he didn't notice very well. At the beginning, he went back to the lab and take out the plate and realized that around the penicillium, E. coli was not able to grow. That was the first idea to arrive to the penicillin. But this is again a matter of causality. So we rather prefer another approach. We rather prefer the modern drug discovery approach, where we are the fishermen and we go to look for molecules that can interact very specifically with the target. So our idea was to generate a library of molecules, in this case of peptides, but they have the capacity to interfere, to interact very specifically with one bait, with the worm. And if you go to fish, if you go to fish in a pond with just 10 fishes, the probability to get back home with something is low. It already increases of 10 times more if there are 100 fishes. But what we wanted to generate, it was a system very specific, like this key. So the idea is to get like this key that get into the door, and only if it interacts very, very specifically with the bait, the key lock, we can or block definitely or open. But again, we had the problem to go to fish. If we want to be effective, we needed to go to fish, not in a pond with 100 fishes, but with millions of fishes. So the pressure that we felt at the beginning of the project was to be able to generate in few months, actually more or less 1 year, a library that contained millions of fishes, like in this case. And we were, I must say that at the end of the project, we have been able to do that. And the system that we used was the yeast to hybrid assay. It's an in vivo screening because we run it using yeast cell. This is just Saccharomyces cerevisiae cell, so our test tube, our lab test tube system. But every one of us knows very well this small fungus. It's used for bread preparation, wine preparation, but again, is extensively used in the lab since decades. So we run yeast to hybrid approach. As the word to hybrid says, we are running with 2 proteins, but these proteins are chimeric. The system uses the yeast cell transcription and translation tool, and we generate 2 chimeric protein, fused, the bait, so the one that we use to go to fish, remember this approach, fused to the GAL4 DNA binding domain. What is GAL4? It's a transcription factor of yeast that is needed to yeast cell to use galactose when it's present. Yeast definitely prefer to grow with the glucose like us, but if there is no glucose, it's able to switch maybe on galactose if present. And that's the goal of GAL4 transcription factor. As any transcription factor, GAL4 has a DNA binding domain and an activation domain. Its goal is to bind the DNA and to activate something else. So to do the DNA binding, it needs a part of the protein that is evoluted for DNA binding recognition, and then there is a GAL4 activation domain to interact with the RNA polymerases. So to debate, we have fused, to GAL4BD, sorry, we have fused the bait protein we were interested, and we put our fishes, our million of peptides, fused in frame with the GAL4 activation domain. Only if the fish recognizes the worm, then there is interaction, and the GAL4 transcription factor is again reconstituted and can activate something downstream. These are the type of screen that we do in the lab. This is a control plate, this is the screen. If we activate the reporter gene, we can only grow in very specific media those cells where the interaction occurs, and all the other that are present in the control can be killed and eliminated. So that's the reason why we liked this tool. First of all, it's in vivo. If some peptides are toxic, it can be eliminated. That's a further step. We save a lot of time to not characterize peptides that might turn out to be toxic later on, and we can simply select for those able of the interaction, and all the rest is killed. We have generated two libraries, one of linear peptide that needed to be inserted in a specific scaffold, and then talking with Sara Pellegrino and the Department of Chemistry, we reasoned on the fact at that time that maybe it was better to select or to generate a library of cyclic peptides because they have less conformation frame. So this is the second library that we generated with the cyclic peptides, our fishes that are here. The library has been published. The first author is the PhD student that did the work with me, and the second one is Andrea Tagliani that is here in the audience. So Stefano now is in the United States. And this is how the libraries look like. So you must think that this is one plate of several hundreds of plates that have been scratched and collected and stored at -80, and now they are ready to be used. For each screening, we can use about 120 microliters, so we have in our SMANO 82 library that can be used in the next year for many other screening. So it's really a huge presence of fishes because, as the title said us, we can screen 4 million of independent peptides and in vivo. And a further advantage of this methodology is that we do not need any conformation information about the bait. So we just need to know which is the gene, and this has been told us by VIBAF group. We simply clone the gene and we went to screen. We do not need any prediction of the structure or crystal of the structure that is even worse. The cyclic library worked very well, and we were quite proud that, about our library because we could produce something that was better of a previous library published almost 14 years ago. We had much more in-frame clone because we improved a lot of the protocol, and we have been able to reduce more than them the presence of codon stop, so enlarging the final title of the library. What we have done in the frame of the NoPEST, we have done about 25 screening. We have dealt with more than 3,000 of interfering peptides. About 400 of them have been characterized, so the group of the pathologists of Silvia Tofolatti did a great job. And among them, about 90 showed a good antimicrobial peptide. We have a library, so we have been able to activate the project work, so the pressure in end disappeared because we were also the group that somehow stopped earlier to work. But we can also move this technique, and for fun or for curiosity, we have done, because we have, for instance, isolated 1 or 2 peptides that can have anti-COVID activity, but also we have screened in collaboration with other colleagues, peptides that might have anti-malaria activity. So what we have generated for sure will not finish with the project, but might be used later on. Before to finish, a special thanks to Stefano, that was the PhD student, and to Andrea, that with me made most of the job related to the library construction. And thank you all you for the attention.
Okay, thank you, Simona, for showing us the technology. So as we have seen towards this is2 hybrid assay, we can isolate several peptides with antimicrobial properties. And so that actually allow us to address one of the main objectives, identify a technology that in few months would result in several candidates, in several peptides with antimicrobial properties to be tested in the laboratory with the assay that Professor Silvia Toffolati developed, and later on, thanks to the collaboration with our industrial partners at Camoxon, to verify whether they are also working under field condition. And we will hear very soon about them. So I don't know whether there are questions now on the technology. Otherwise, we might have a final discussion on the different aspects. And so now we go to the next talk, and here we try to address another important aspect of these peptides. You know, they are a natural protein made of natural amino acids, and the idea was that they might be not so stable once we are under real field condition. And that's the reason why we asked Professor Sandrine Ongery from the University of Paris-Saclay to try to look at our peptides and eventually develop strategy to make them more stable and also more efficient versus our target proteins. And Sandrine will now report about the so-called peptidomimetics that she developed in the frame of the NoPEST. Sandrine, the floor is yours.
So thanks a lot to Paolo, of course, but thanks a lot for the invitation here in this beautiful academia and room. So I will try to explain to you how we design new derivative of peptides to increase their stability. So first, what is a peptide? So peptides are short chain biomolecules of between 2 and 50 amino acids. So they are smaller than proteins, and they are linked by peptide bonds. So peptide bonds are formed by CO and NH atoms to form this bond between amino acids, which I represented here in different colors. So most natural peptides are from animals, plants, and microorganisms, and peptides are ubiquitous in living organisms, and they modulate many physiological processes. So natural and synthetic peptides can be produced through chemical synthesis, but also through biological fermentation or even gene recombinations. Okay, so I come from pharmaceutical fields, so I love peptides for their therapeutic advantages. In particular, they show high power. So drugs, peptide drugs show high power and high effect at low doses. They have also the behavior to be very selective. So they show less side effects and a better safety because they are degraded into their constituent amino acids. First of all, they are also very good for the environment because they are degraded, so they do not accumulate in soil and water. They have some disadvantages, so it's why we try to modulate a little bit and to modify slightly peptides, it's because they have short duration in vivo and they cannot be administered orally. You can see here how the last century, during the last century, pharmaceutical companies were afraid by peptides because of their short duration in vivo. While now you can see that almost 80 peptide drugs have been approved and they are available for therapy. You can see here the increase of peptide drugs approvals during the last decades. More than 200 peptides are also in clinical development and 600 peptides are also under preclinical studies. So this is a typical peptides which show very nice activities and biological activities. However, why we are afraid by these peptides, especially in vivo, because many proteases can cleave these peptide bonds, giving small peptides or amino acids which are not anymore active. So chemists need to modify slightly some peptides to try to increase their stability in order to be used at least few hours in vivo or stable few hours in vivo. Sorry. So here I show you few modifications of these peptides to stay biologically active, but to be more stable. Here you can see that we just modulate one amino acid by replacing a carbon by a nitrogen, or here by increasing the size here of the amino acid by one carbon. You can also slightly modify the side chain modification, so increase here the size of this lateral chain by replacing an isopropyl by a cyclohexane, or modifying here the chirality of an amino acid passing from a L amino acids to a D amino acids. Cyclization is also a very good way to increase the stability of peptides. So here you can see few examples of peptides used in clinics. Of course, insulin, which is a completely natural peptide, and also this anti-diabetic peptides, exenatide. So they are composed by only natural amino acids. However, they cannot be used in vivo. So we need to modify slightly some peptides. For example, here, abar helix has been modified in the backbone, but also in the lateral chain to obtain more stable and very active compound as prostate anti-cancer. Here also we have very nice examples of modification of a small peptides, semaglutide, to be used in vivo. So this is the first example of peptide as anti-diabetic peptides used in vivo just by modifying two amino acids, substituting by a fatty diacetyl chain, and adding a compound that enhance the absorption of this peptide. So semaglutide has been approved to be used orally by the FDA in US in 2019 and in Europe in 2020. So you can see that peptides in therapeutics can have a very high range of activities, and now they can even be used in vivo. What about peptides in eco-agriculture? Here you have this review showing that peptides are increasing and have increasing interest as new tools for plant protection in eco-agriculture. Paolo showed you already this graph, but you can see that from the last century to this century, the number of peptide publication has increased. And even now, some of them are commercially available because to date, 18 peptides have been commercialized as green agents for plant protections. So the idea to use this peptide is for the environment, because they are expected to have no toxicity for other species, they are degraded, and they do not accumulate in soil and water. They have been applied as antimicrobials and immune inducers, as plant growth regulators, insecticides, herbicides to protect plants from bacteria, virus, pests, and weeds. So among these 18 peptides, I show you here 2 examples of quite large peptides. You can see here the bioinsecticide Spear, who won the Presidential Green Chemistry Challenge Award in 2020, and the Best New Biological Agent Award in 2021 in the USA. This large peptide comes from a spider venom, and it blocks 2 ion channels in the insect nervous system. So this compound can be used with other products to prevent the emergence of resistant insects. So you can see here that the interest of peptides in eco-agriculture is increasing, and in the field of NoPEST, we were working, of course, with Milano, and Milano, for example, showed this peptide, cyclic peptide, very interesting to modulate the galactose metabolism in yeast by interfering with GAL4 transcriptional activity. So from this peptide, we worked in the frame of this project, and the idea was to target oomycetes that cause mildew. And why I was very interested in this field, it's because conventional fungicides as copper salt the use of copper has been shown to be very harmful to animal and human health, such as promoting neurodegenerative diseases. So even if I come from pharmaceutical fields, I was very interested in this agriculture field because of the toxicity of the copper salt. So as Simona told you, and also Sylvia told you, we discovered in the NoPEST project 2 kinds of peptides, cyclic peptides and linear peptides, which were as effective as copper salt, but also they didn't show any toxic effects on non-target organisms, human cells, bacteria, Daphnia, or bees. And you will see that Sipcam Oxum test them also in fields, so they will give us some interesting results about these peptides. But the problem with these peptides were for the cyclic peptides, the synthesis, which is quite difficult if we want to produce them in large scale, and the fact that linear peptides can be not very stable in fields conditions. So the objective in my lab in Paris-Saclay was to try to modify very slightly these linear peptides in order to increase the stability toward proteolysis, but to keep the larger number of natural amino acids as possible, so only modify 1 or 2 amino acids, and to have an easy synthesis or chemical synthesis or biological fermentation synthesis. So we replace 1 or 2 amino acids, natural amino acids, by D amino acids, just changing the chirality here, or introducing ASA amino acids or hydrazine acids. And the results were very good, because if we insert only 2 D amino acids in a small sequence of these peptides, we kept the antimicrobial activities while we dramatically increased the stability towards proteolysis. You can see here that the natural peptides was completely degraded after 10 minutes in this pronase assay. So this pronase assay contains a mixture of proteases. while by introducing 2D amino acids, we dramatically increase the stability. Even after 2 hours, you can see that we still have the compounds. These peptides are also quite easily prepared because automatic synthesizer can be used, or even we can plan the ribosomal incorporation of the amino acids. We modify many things, but the one of the most efficient way was to also insert one hydrazino acid. So by replacing more or less in the middle of the peptides, one L amino acid, one natural amino acid by this small hydrazino acid, we kept the antimicrobial activities while we increased the stability towards proteolysis. You can see here, the natural peptide was degraded after 10 minutes, while we kept some peptides even after 2 hours. So for this work, I would like to thank a lot my two collaborators, because in the frame of this project, 2 postdoc researchers were recruited, Lea Radal, who stayed two years, and Alice Della Valle, who stayed 1 year. And they both found a very good job after the training in our lab and in this project. Lea was recruited by SB Peptide Company in France, and Alice Della Valle was recruited at IRBM Company in Rome, in Italy. And I would like, of course, to thank all my collaborators in the frame of this European FET-OPEN NoPEST, and especially the team of Milano, who prepared some natural amino acids, Lucia Feni and Sarah Pellegrino, and the person who tests these compounds. And I would like to thank you for your attention. Do you have any questions?
Thank you, Sandrine. I think the one question would be how expensive is to produce those peptides chemically and containing your modified amino acids? That's probably the most obvious question.
That's a difficult question. Of course, peptides are quite costly, much more than copper. We cannot fight against this fact, of course, but you can see that even in pharmaceutical company, they found peptides, even if they are more expensive, they are so specific, so you don't have to spread, I hope, in the fields. Maybe Sipcam Oxon will answer better, but we should not have to spread too much peptides. So it's difficult to say how it costs, especially kilograms of peptides. But maybe you have the answer, because I think we try to produce some peptides, no? Or Sipcam Oxon has the answer. It's costly, but I think it's very specific, so you don't have to spread much these peptides.
And this eventually can be done in combination with precise aggregation tools. And so Javier might tell us in a few minutes eventually whether that is possible. Please, Professor Alpi.
At the end of your talk, I was following the specific work done with protease. This mixture of proteolytic enzyme contain both exopeptidase and endoproteolytic enzyme.
Yes, they contain a large range of proteases, because we don't know in plants, and it will be depends of the bacteria around the plants and all the fungi around the plants. So it's difficult to predict which proteases. So it's why we choose a very large range of enzymes.
I suppose from the work done and with completely different object. But the endoproteolytic enzymes should be the ones that work mostly in comparison to the exopeptidase. Do you agree with
Yes, yes.
Okay.
Okay. Okay, if there are no more questions, I think we can proceed with the next speaker. So you can imagine when we started this Thanks a lot, Sandrine, first of all. Thanks a lot. So you can imagine when we started the project, thinking about peptides, basically from a simple idea from scratch, we thought that we should have also another strategy that could go on in parallel to the identification of peptides. And actually, we could use the identified peptides to identify a different kind of molecule, what we call it non-peptide small molecule. And so we gave this task to Professor Anok Senderovitz from the Bar Ilan University in Israel. And Anok had the task to both take the target protein that we have identified thanks to the VIBAL studies, and looking at the protein, looking at the structure of the protein, try to use it for in silico screening and see whether we could have some non-peptide small molecule that could target those enzymes identified by analyzing the cell wall. And in addition to that, we asked Anok also if, based on the peptides that we have identified showing an antimicrobial property, if we could use the structure of these peptides to identify a natural molecule that could mimic the activity of those peptides. And that has been the task that Anok has carried out during this 6-year project. And we are now happy to listen the results that Anok has obtaining during his activity. Thank you, Anok, for being connected with us from Israel. I leave you the stage, and we are looking forward to hear from you.
Thank you. Can you see my presentation?
Yeah. Yes. Now, yes.
Okay. So I am really sorry that I cannot be with you right now. Looking at the room you are in, it's much nicer than the office I'm in now. So I have to say that I really envy you. Anyway, I'm going to explain a little bit about what virtual screening is all about, and then I'm going to move and talk about the virtual screening in this project. And one more thing before I continue. Okay, and I hope you can also see my pointer now. Okay, so what virtual screening is all about. So today, it's actually a very mature paradigm in computer-aided drug design, much less so in agriculture. And the idea is to use computational techniques in order to screen very large, and I'll say in a minute how large, compounds library, in order to find those that are most likely to bind to a specific target. Now, the results of virtual screening should always be validated experimentally, and this is actually what we have done also in this project. So there are two main components in virtual screening. The first one, it's all about the target. We need to identify targets, relevant targets, and then we need to identify compounds collection, which we can screen against these targets. We are typically using what is known as a funnel-based approach, which means we are using kind of cheap computational methods at the beginning, and then we are using more expensive computational method at the end so that we can quickly filter out very, very large compounds library. So there's two main methods to virtual screening. The first of them is what is known as target-based, which looks at the target, and the other one is based on ligand-based, looking only on the ligands. So target-based virtual screening is what we mostly applied in this project, and the idea is the following. You take a protein, either a structure. If you don't have a structure, you build a model of the protein. You then take a virtual library. Now, this library is virtual in the sense that you have it only on the computer, but at the same time, these are libraries of compounds which can be either synthesized or are already in stock. And today, there are many vendors who would sell you many compounds library. You then screen them, and we're using a combination of pharmacophore models and docking. I'll explain briefly what they are. You get the virtual hits, you then assay them, you get what is known as lead compound, and then you try to optimize them, again, using computational techniques. So where do we get the structure? The first source of structures is the PDB, the Protein Data Bank. And as of, I think, yesterday, there are over 200,000 structures in the PDB and over 1 million models. And this is just telling you the growth of the PDB over the years. Now, if you don't have a structure, then you should build a model. And this falls under the category of homology modeling. As some of you may heard about the AlphaFold method, which is now the best homology modeling method which exists. Unfortunately, when we started the project about 6 years ago, this method was not around, so we used other method. The main idea in homology modeling is that if you have two proteins which are very close at the sequence level, then they should also be very close at the structure level. And by close, I mean 30% sequence identity is usually sufficient to generate a homology model. And homology modeling is a whole area of research, and there are ways to do it, and we know how to do it. So I'm showing you here just as an example, a cartoon of multiple sequence alignment, which eventually lead to a protein structure. And in a few minutes, I'll give you a more precise example, which has to do with the structures which we modeled in this project. Where do we get libraries? So today, there are many vendors, which are shown here, that actually produce a huge library of drug-like molecules. In particular, Enamine is a company which has the real space. And the real space contains today billions of compounds which could be easily synthesized. These are not commercially available at the moment, but Enamine is committed to synthesizing any compound from this database within 3 weeks, and they are usually up to the task. Just as two anecdote, drug discovery companies have proprietary libraries which are much larger. So the Merck collection is today 10 to the 20. And this small green dot over here is the number of approved drugs, only about 10,000. And this is like 3 years old slide. But the point is that there are commercially available libraries which we can easily download to the computer and screen. How do we screen? So we use the concept of a pharmacophore. And the idea behind pharmacophore is that it could be regarded as the highest common denominator of a group of compounds which have a certain activity. Pharmacophores are basically or typically defined in terms of several functional groups. So hydrogen bond donor, acceptor, positive charge, negative charge, hydrophobic moiety, and aromatic moieties. So if a number of compounds have a certain arrangement of pharmacophoric group common to all the active compounds and absent from the inactive compounds, then we can basically say that this arrangement of functional group is the pharmacophore. And if we are able to identify this specific arrangement on other scaffold, then these other scaffold may also be active. And this actually comes from the similarity principle idea in chemistry, which suggests that if you have two compounds which are structurally similar, then they're likely to also have the similar activity. And under the concept of pharmacophore, similarity is measured in terms of the arrangement of pharmacophoric features. Now, the best way to generate a pharmacophore is if you have a crystal structure of a protein in complex with a small molecule, like in this case. And if this is indeed the case, then by 2 clicks of a button, you can generate a reliable pharmacophore model, and then you would be looking for other compounds which have this arrangement of functional groups. Now, if there is no, if we don't have such luck, if we don't have structure with ligands, then we can take a binding site, and we can look at the amino acids at the binding site, and from the binding site, we can generate a pharmacophore model. So, for example, if we see in the binding site something which is negatively charged, like here, then our pharmacophoric feature will be something which is positively charged. If we see something aromatic in the binding site, then we will put next to it an aromatic feature in our ligand. If we see something which is positively charged, we'll put against it something which is negatively charged. So using a structure of a binding site only, we can again generate a reliable pharmacophore model. I'm going now. There is also a way to generate pharmacophore models based on ligand alone. And as Paulo said before, we use this strategy to look at peptides, generate pharmacophore models just from the peptide without knowing their precise target, and screening compounds. I'm not going to talk much about this because while we have screened the compounds and we have a list of compounds ready for actual biological assay, these were not validated experimentally until now, so we don't have validated results for them. But I will show validated results for a screening which emerged from a binding site. Now, pharmacophore screening is very quick, and we can screen millions of compounds within a matter of weeks. However, you get too many hits. You can get 1,000, 2,000, or 3,000 hits, and this is way too much for us to test experimentally. So what we do is we filter the hits using more advanced computational techniques, and basically we're using what is known as molecular docking. And the purpose of docking, or docking has 2 purposes. The first one is to obtain reliable binding mode of ligands within binding site of proteins, and the second one is to get at least approximated binding free energies. Now, we don't get completely accurate binding free energies because in order to be able to screen thousands of compounds in reasonable amount of time, we need to do some approximation in the way we score the compounds in the binding site, yet we are able to get nice hits. Now, the docking of a molecule into a ligand is actually a very complex process because what you actually need to do is model computationally this process. You have a ligand which is floating or swimming in solution, and you have a protein binding site where the binding site is also you typically fill with water molecule. And what you need to do is being able to model the process in which the ligand binds to the binding site. And there are a lot of contribution to this process. Desolvation of the ligand, desolvation of the binding site, conformational changes of both the ligand and of the binding site, entropy effect emerging from the fact that you restrict the movement of the ligand in the binding site, and so on and so forth. So docking is not it's not a computationally easy task, yet there are quite a few programs which can do it very efficiently. So what do you need in order to do docking? You need to identify a receptor binding site. If you have a complex with ligand, you can do it from the complex. If you don't, then you have to search for binding site using, again, computational algorithms. Then you need to place the ligand in the binding pocket in many, many different orientation and internal conformation of the ligands. And then you need to compute the interaction, which can approximate the free energy of binding between the ligand and the receptor. And you do it for many, many conformation of the ligands. You can also introduce many conformation of the receptor, but this will cost more in terms of computational effort until you are satisfied with the result. There are many, many docking tools and many docking. So there are many tools which are able to place a ligand in the binding site. So this is this stage. And there are many scoring functions which can perform this stage. that is to score the ligand in the binding site, and there are obviously many combination of them. We typically use a program called Glide from Schrödinger, which has several scoring functions. So we start with the more simple scoring function, and then we subsequently go to a more complex scoring function, typically ending with also introducing flexibility into the protein structure. So this was kind of a general introduction. Specifically in this project, we worked on 2 proteins. We worked on more, but I'm going to present the results from 2. So the first one is pectin metylesterase. So cell walls of plants are made of pectin. And pectin metylesterases, at least pectin metylesterases of plants are actually essential for remodeling the cell wall of plants. However, oomocytes also use pectin metylesterases in order to catalyze the first step of the oomocyte invasion into plant's tissue. So the idea is that if we are able to inhibit the pectin metylesterase of oomocyte without touching the pectin metylesterase of plants, then we may have a green pesticide. Now, fortunately for us, the sequence identity between pectin metylesterase of oomocyte and plants is very small. So we really think that if we develop something which is specific for the oomocyte, it's not going to affect the plant itself, which is what we want. So we got from our colleagues in Sweden sequences of pectin metylesterases of oomocyte. We looked into the PDB, and we found that while there is no structure of the pectin metylesterase from the specific oomocyte, there are enough structure, enough sequence identity to allow us to model it. So this is, I'll show you the models, I'll show you a more refined model in a minute, but this is a structure of a pectin metylesterase from one of the plant pathogen. This is actually what it does, and this is the mechanism of its action. It basically removed an esteric moiety from this position over here, replaces with an acidic moiety, and by this it catalyzes, as I said, the first step in oomocyte invasion. Now, there are several amino acids which are in the binding site where all this happens, and this helps us to identify the place or the exact binding pocket in the pectin metylesterase which we want to inhibit. So we did the sequence alignment between the PDB structure and the pectin metylesterase from the oomocyte we wanted to model. And what I'm showing you here is superposition of the structure of the template from the PDB and our model. And what I'm showing you here on the right-hand side is one way in which we validated the model. So when you perform homology modeling, you need to validate it at 2 levels. The first level, you want to see that your protein looks like a normal protein. And this process validation over here, without going into too many details, demonstrated to us that this is indeed the case. The other thing you need to make sure is that your protein looks like the protein you wanted it to be. And for this, we're using this metric. So this tells us how much our model is similar to our template, that protein on top of which we modeled our sequence, and 0.5 angstrom is a very good fit, which basically means that our model looks like pectin metylesterase. The next thing we did is the following. In the PDB, there are several structures which also contain the pectin chain over here, which is being catalyzed by the pectin metylesterase. So we took it and we moved it into our model. So what I'm showing here is our final model, pectin metylesterase from the oomocyte together with part of the pectin chain. And these 2 residue over here, these 2 aspartic acid, if I'm going back to here, these are the aspartic acid here and here, which participate in the removal of this ester moiety. So we know that roughly here is where we need to inhibit the protein if we want to stop it from destroying the pectin of the plant cell wall. We took this structure and using pharmacophore approach, we turn it into a pharmacophore model, which is shown here. So these are hydrogen bond donors and acceptors, and we have like 8 pharmacophoric moieties. We then took about 43 million compounds from different vendors, mostly enamine, also zinc and molport. And these 2 small libraries are libraries of FDA-approved drugs. The idea was we wanted to see if there is anything within the FDA-approved drugs which would fit the pharmacophore model, and this is called in computer-aided drug design, this is called drug repurposing, using an old drug for a new target. Unfortunately, in this case, we didn't get any hits from this, but we thought we'd try it. So we screen 43 million compounds. We only took compounds which matched between 6 and 8 features of this pharmacophore model. So these are the best fitting compounds. And there were about 7,000 of these compounds. We then docked them into the binding site using one docking program. We got 728 compounds. And then we looked at them and we decided that we should dock all of them using yet a more sophisticated docking program. And we ended up looking at about 700 structure on the screen. So the final stage of any virtual screening is 2 people or 3 people sitting together and just looking at one compound after the other in the binding site. It turns out that there is really no substitution to a trained computational chemist who is actually looking at the compounds. And after spending hours looking at the compounds, we ended up selecting 21 compounds and sent them to the Milano team. And the Milano team were able to buy, I believe, 11 of them, did the experimental work, and the results are shown here. The results, these compounds are not as active as the peptides. On the plus side, they're much cheaper than the peptides, and they're optimizable, or we think that they're optimizable. So several of the compounds actually, especially this compound number 4, worked out pretty nicely at even 100 micromolar, and the 2 replicates were actually very nice. So we have some, I don't know, oil signs, some initial points to work on. So what did we do next? One more thing. We took 8 compounds. 3 of them were inactive, 5 of them were active, and we docked them again into the model. And we saw 2 things. The first thing we saw is that the docking score is able to pretty well differentiate between the active compound and the inactive compound, with one exception over here. This is an active compound with a poor docking score. But aside from these compounds, all the active compounds has better docking score than the inactive. In docking terms, more negative numbers suggest better binding. More than that, what you're seeing here is superposition of all the active in red and all the inactive compound in the binding site. And you see that all the active compounds are binding roughly in the same place and in a similar manner, whereas all the inactive compounds are spread all around the binding site. So this gave us a good feeling about it. More than that, some of the active compounds actually contain several OH groups. Now, I'm just going back to this slide. This is a pectin moiety, and a pectin is a multi-sugar, which contains many OH groups, and this pectin moiety was used to build part of the pharmacophore model. So the fact that the screening was able to retrieve compound with OH group, to us was a good sign. So we took all these compounds and we docked the docking mode, and then we built pharmacophore models from them. So we built 5 pharmacophore models from the active compounds, and we also built 3 pharmacophore models from the inactive compounds. And we again screened about 40 million compounds against the 5 pharmacophore models. We took all the compounds which turned out to be active, and screen them against the pharmacophore models which were built from the inactive compound. The idea here was, if we get false positive in this screening, maybe we'll be able to get rid of them in this screening. We then looked at all the compounds which make interaction with these two residues. And we also looked at all the compound with binding energy smaller than this number. Why this number? Because this number was the number of the lowest inactive compound. So we were only looking for compounds which have higher or better docking score than the inactive compounds. We then based our final selection on compounds which has a docking score smaller than 9.3, which was the best compound over here. Several interaction with the aspartic acid and with additional amino acids, and we ended up with 37 compounds which were sent to the Milan team. My understanding is that some of them were already tested and are showing encouraging results. So this is the story of the pectin metylesterase. In parallel, we also worked on another protein, which is PEX RD54. And this protein, which is shown here, is binding to another protein, which is called ATG8. And in doing so, it kind of hijacks the plant immune system. So the oomocyte hijacks the plant immune system for its own benefit. So again, the idea was that if we are able to inhibit the interaction between this compound, between this protein and the ATG8, we will maybe be able to develop the beginning of a green pesticide. Now, this is a more complex problem because here you need to interfere with protein-protein interaction. And using small molecules to interfere with protein-protein interaction is something which is rather complex. Fortunately for us, we found in the PDB this structure. And this structure is the ATG8 with the 5 amino acid from these compounds bound in a complex. So this is actually a very good starting point for a pharmacophore model. So we generate a structure-based pharmacophore model, which is shown here and here. Again, we took these 43 million compounds from the initial database and screened it against the pharmacophore model, did again the docking with Glide XP, and then introducing flexibility into the protein structure, looking at about 200 structure by eye, selecting 19 of them, and these 19 were also sent to Milan. These were not tested yet, but at this point we have 19 compounds which are ready to be screened in an assay against this protein. In parallel, we also have a list of compounds which were screened using the pharmacophore approach based on the active peptides. But again, since they were not tested yet, I'm not showing them. So, in conclusion, as I said at the beginning, virtual screening is a mature paradigm in computer-aided drug design, much less so in agriculture, but I think it has a great potential. Using virtual screening, we were able to find some hits, and we were also able to start their optimization. Again, at this point, they are not as active as the peptides, but as I said before, they are cheaper. And we know, I mean, if we need to proceed with further optimization, we know how to do it. We're using computational tools to do it, but we know how to do it. And as I said, we have a list of compounds ready to be tested for other targets. And I have to finish by thanking, except of course, all the the member of the consortium, the people in my lab who did the work. So the work was done primarily by Natalie Kazanov. She is my lab manager, has been working with me for the last 15 years or so, and by two talented master students, Lina and Shachaf. And thank you very much for your attention.
Thank you, Anok, for this very nice presentation. I would like to thank you once again because of the difficult moment you are living in your country. So unfortunately, during these 6 years, we didn't have the possibility to have a meeting in Israel, although it was initially planned. I have a question for you. Is there room to transform those candidate molecules that you have identified into natural products? In other words, do we have the possibility to identify natural molecules that are close in the structure to the candidate that you have identified? What is the probability of getting it? I'm telling you this because our industrial partner is always stressing the fact that if we manage to get natural molecule, the preparation of the dossier and the cost of registration will be much, much lower, if I'm not wrong.
So I don't know what the probability is, but yes, we can certainly try this. So there are quite a few databases today of natural product from many sources. It is reasonably easy to look at all these compounds and search, take the most active compounds and search for close analogs in databases of natural compounds, look at the most similar one and test them, dock them and test them. We can also do another docking. So this docking study focused more on drug-like compounds, but by the same token, we can do the same procedure on databases of natural occurring compounds. A potential problem I see there is that some of the database indeed contain natural compounds, but not always how to get them. But in principle, it's technically certainly very feasible to try to identify natural products which are similar to these compounds.
Thank you. Thank you, Anok. I don't know whether there are other questions. Probably not. I thank you once again, Anok, for all your work.
My pleasure.
And now we can move to Javier. As we said, peptides and even some of the molecules that Anok identified can cost a lot. And so one possible solution is to spray them at the very beginning of the infection and specifically on the place where the infection is starting. And for doing that, we need the sensor. We need the precision agriculture tools. And that was actually the main task that we gave to Javier, to develop a sensor working under real field condition that could detect at the very early stages the infection from Plasmopora viticola and Phytophthora infestans. Thank you, Javier, for all the work that you have done. I leave the floor to you.
Thank you, Paolo. Everybody, good afternoon. Now we are moving to precision crop protection. So we are going to explain to you how we can detect the main diseases that we are talking about this afternoon. So can we detect the downy mildew and also Phytophthora infestans in potato and in underfield conditions? So in our team and Televitis group and the research group at the University of La Rioja in Spain, we have been working for the last 20 years in precision viticulture. So it means that we use the data to improve the decision-making process, in contrast with the traditional conventional agriculture based on visual inspection and the traditions. So 3 years ago, we published this review where we show in this review that we can use the new technologies, new approaches for improving the agriculture decisions. So we try to promote a more efficient and more sustainable agriculture. So in Europe, we have the Green Deal frame. This is a good frame for everybody. And from the farm to fork strategy, as my colleague mentioned before, one of the pillars is to reduce
the pesticide use. In some preliminary studies, our colleague in Apple Orchard showed that with the early pest disease detection, the pesticide use can be reduced until 40%. That is very, very important. And we try to follow this way in viticulture. So in the goal or the main task in this work package was to develop new tools, new strategies based on new technologies for detecting the main diseases as red vine downy mildew and potato leaf blight. So in the first 2 years, we worked under lab condition. That is more easier way. But now I am going to explain you the activity to detect this disease under field conditions. And to detect this disease, the first symptoms, it is not very easy task because we are talking about, the symptoms of downy mildew are very small. We are talking about the 2-3 square millimeters in the very complex scenario in the canopy with a different illumination condition with the dark leaves and also illumination leaves. So this is very, very easy task, but we try to just to identify the first symptoms of this key disease in viticulture. So we ran a 4-year experimentation work in Spain, and with different, we work with different grapevine variety, with different trellis system, with different, we use different images with thermal and RGB images in this way. We use also different platform for image acquisition. We use a manual sensor, and also we develop a full round phenotyping platform for on-the-go image acquisition under field condition, and also we try to use drones for image acquisition in vineyard and potato. So I mentioned we develop a very powerful ground phenotyping platform for taking on-the-go image acquisition, thermal and RGB images, and we use the GPS in order to positioning all the images, in order to get all the images where we know the real position in the field. And so for downy mildew detection, we use different approaches. The first approach is the use to convolutional neural network for detecting downy mildew symptoms in the field and localize these symptoms. And the second approach, we use semantic segmentation for counting downy mildew spot. That is not easy. And the third one was they used YOLO is a new algorithm for counting downy mildew infected leaves with downy mildew. So just to summarize the main result for the 3 different approaches, here you have a summarize the summary of this methodology. We use this convolutional neural network that they show a good result for computing images in other fields, and try to use this deep learning algorithm for detecting the first symptoms of the downy mildew. And we use different architecture for the different neural networks, CNNs, and also vision transformer. And here in the green rectangle, you have the highest performance architecture called efficient network X26 to X for this is the best model, and we got in this case. And for visualize the result of this architecture, we use this approach they call red cam application to visualize how this neural network can be detect the symptoms in the field, in the leaves. And here you have the difference, the behavior of the original images in the first column, and also the behavior of the visualization from the different architecture. I'm sorry. And for validation, we use the confusion matrix approach. And here you can see the result In this case, here the predicted class versus the actual class of the downy mildew. And for the downy mildew, higher than 65% of the images were correctly classified. With the no downy mildew class, yes, we got until 94% of the images were correctly classified. And even this model performs well under different conditions for different platform, for different sky illumination condition, and even for the different grapevine varieties. And we published just this paper a few months ago. And just to highlight, I'm my PhD student, Inés Hernández. She just finished the PhD for downy mildew detection in 2 months. So that is, she is doing a very good job in this topic. So the second approach we use with semantic segmentation. This is a new deep learning algorithm. It's very, very powerful. It shows good results in another topics, but it requires a lot of time for labeling, because every pixel of the image have to be labeled and classified. So it requires very long time for labeling all the images. So here also, in order to increase the variability and the robustness of our model, we did a lot of job with the data preparation to rotate, to zoom the images in order to increase the variability. And here just one figure about the results, and each point represents one different vineyard. And here you have the relationship between the number of real spots versus the number of predicted. So we have a good correlation. So it means that the models work very well under different conditions with different vineyards. Even I wanted to highlight, even when you with a lower very low spot, it was very well. So it means that we can detect the first symptoms in the vineyard with this approach. Just we try to use a similar approach than the pathology used in the field, just to count the number of infected leaves using a YOLO. This is a new deep learning algorithm using DNA approach. Here this is more rapid for labeling, because you have to be only to label the whole leaf. So this is more rapid to label and to process the images. This is the algorithm. The idea was very simple. Just here you have the ground truth on the left, and here is the YOLO detection. So just to detect the infected leaves in the canopy, in the vineyard canopy. So we work to improve the methods and the different variety with different skylight condition and daytime and also at nighttime with Tempranillo and Muscat in Spain. And here we have the result. And just we saw this is the correlation, a good correlation indicates that we can count automatically the infected leaves in the field, infected leaves with downy mildew. That is very, very important in order to get a good diagnosis of this key disease and the field condition. So here you have a representation about what is the picture about the one vineyard with the low severity downy mildew. Only 60% of the leaves are infected with downy mildew, and this is the high severity vineyard with downy mildew. So almost 70% of the leaves were infected with downy mildew. So this is very difficult. Different vineyard with different incidents on downy mildew, and also in this way we can obtain the severity. Here you have a good representation of different vineyard with different downy mildew severity. This is the relationship between the total Also, we count the total visible leaves, that is very important, and also the mildew-infected leaves. So we can get the ratio between the both parameters an easy way. So this is a good way to dynamically diagnosis in underfield conditions. So of course, as my colleague mentioned before, we try to be more efficient. So maybe to apply these more expensive molecules more efficiently in real conditions. So we need to map the results, this diagnosis in the field. So we try to do that because I just highlight that with this our phenotyping platform, we use the GPS. So all the images and we got were georeferenced, so we can map easily the results. We can get this type of the map down in mildew incidents with the different incidents with different zones, so we can apply different doses for different areas with different incidents. That's the idea. So we can save a lot of pesticide. Yeah, that's the idea. So even we can do the plant by plant diagnosis if it is needed. So this is it is a good result for precision crop protection and to localize the spray systems. So just to finish, I would like to also to show you the further sulfur in potato lead blade to detect the phytophthora infestans. And so also we run a 3-year experimentation setup. Here you have the summary. Oh, sorry. So we use also different platforms, ground and drones. And we use, in this case, convolutional neural networks. And we use a sliding window. It means that the whole image was divided by the sliding window in order to detect the first signal for any single window. And just we just said that the disease was detected at least with the probability for any single image was higher than 0.7. And this is the summary of the results. In this case, in the first line for the sub images, for the single images, so very, very high result. And for the whole picture, for the whole images, yeah, taking account for all the result for the sliding window, we just reached the 100%. This is the first result that they are very, very high result. We are very, very proud, even with a very small number of images, and we have. And we published 2 months ago this previous work in archive with the same colleague with the University of Granada in Spain. So thank you very much, and you can reach me here or even available. It was a pleasure to be here to work with all of you.
Thank you, Javier. Thanks a lot for your very nice presentation. So, are there questions for Javier? Obviously, the tool you develop looks rather complex. I think a farmer at the moment will not be able to use all these sensors. Can you tell us when or at least predict when a simple tool can be prepared and given to a farmer so that that can be user friendly and it can really use under real field condition?
We are going in this direction, but we just 2 years ago we have developed a new spin-off at the University of La Rioja in order to transfer the whole technology, the whole knowledge to the real condition, to the robot, to the real wine industry. So that's the goal in our team, just to transfer this technology. So we are working to compact, to miniaturize all the sensor in a small we call box in order to set up all these sensors in the tractor. So when the tractor go to the field, they can get the images just for free, in a very easy way, and they can send to the cloud in order to process all the images. And in another way, we are working to develop a more robust model in order to work in different variety, in different vineyards, in different conditions. So this is the idea. So to transfer the technology, but I am very sure that this technology reach the field and to the grower in a very short time.
So I assume in this field, artificial intelligence will play an important role, right?
Sure.
Are you already using this?
We are already using, but now we are working just to develop the sensor, a compact sensor, a not very expensive sensor to mount in the tractor.
Okay.
In order to get a high precision images, because we need the very, very high precision accuracy, high precision images in order to detect the first symptoms. We are working also with another diseases, with SK, etc. The disease.
Okay. Thank you, Javier. Thanks a lot again. Okay, and now we move to our last talk. As you can imagine, we have worked a lot in the lab in developing molecule, sensor, etc. And then we had to test whether all this knowledge, all the tools that we developed were really working under field condition. And that was the task of our industrial partnership, Camoxon, and Maria Isabella Pozzi, that is working for SIP Camoxon. She's going now to show us the results of the field trials that they have conducted. Maria.
Good afternoon, everybody. For me today, it's a very great pleasure to be here and to have this opportunity to work and the results that together with Pietro, we carried out in open field condition. So let's start seeing this evaluation of our peptide that was tested against the grape downy mildew and potato leaf blight in open field condition. First of all, we are talking about the working package number 8, that is regarding the feasibility, delivery, economic and environmental and societal impact, and in particular to the deliverable number 8, implant inhibition of the selected pathogens by reformulated peptidomimetics and non-peptide small molecules. In particular, we were responsible for the task number 8.2, the field test. As you can see in the map, we pointed out with a red mark the location of our test. So as you can see, we tested 2 times here in Spain, just next to Santiago City. One trial was performed here in France, near Bordeaux, while 2 trials were performed here in Turin area, and one just next to Bologna. This blue point is referring to another trials that we performed internally, and it was regarding the first screening of the formulation of our peptides. Okay, so here in the table are resumed all the trials that we performed, and we have some details about these trials, and as you can see, the location, the crop and the target, and the protocol description. One point that is very important is, for example, the inoculation. As you can see, we have in some trials an artificial inoculation, and this is why, because we wanted to, for sure, avoid any risk about disease present failure, and so to be sure to have the good condition in order to better evaluate the peptide performance, we decided to artificial inoculate the pathogen in the field. The rate that we applied, the grams per hectare, here reported, we started from the this value that is 37.5, that is the value about the 50 efficacy dose that was figured out and shared by University of Milan. And we decided to set this value as the lowest rate. Then we decided to double it and triple it and to test these 3 doses in the field. And we applied 6 times these peptides in the field during the trials. Going into details in the protocol, for example, we can start with the grape downy mildew. Here, I just want to focus on this point that we have 3 different clusters. And this is why, because we have enough amount of one peptide, that was the LP20. And so we wanted to test it straight, alone, in the 3 different dosage. And then another cluster is represented by the LP20 peptide plus an adjuvant called adjuvant 1, that was coformulant that we use usually in SIPCAM as a good coformulant for bioproduct in 3 different dosage. And last cluster is represented by the CP20 plus the adjuvant. And as a reference, we choose this kind of commercial product that is a copper sulfate-based product. While for the potato-led blight, we just have the 2 clusters that are LP20 plus the adjuvant and the CP20 plus adjuvant in the 3 different dosage. This is why we finished the amount that was devoted for these kind of trials. Okay, so let's start with the first trial. From this slide on, we will see very in detail every results that are coming from each trials that we performed. So the first one is this one carried out by the Agricoltura 2000, that is the contractor, Italian contractor that ran this trial. And in the graph is presented a type of analysis that is called AUDPC. That means the area under disease progressive curve. In this case, about the leaves situations. So we are not considering a single assessment that was recorded in the trial, but we are considering the disease global expression among all the trials. And this is very important to consider because it expresses, in other words, it's the expression of the product protection level that was achieved in all the trial. So starting from the first, as you can see, we have the highest value achieved by the artificial inoculated untreated. And I mean, this kind of value was an expected results. And this means that the disease pressure in this treatment was very, very high. And it was good. While here, it's the untreated natural, so we didn't apply any kind of artificial inoculation, and this express the natural condition. While here are represented the 3 clusters that we have, so the LP plus the adjuvant, and the CP peptide plus the adjuvant. And as you can see, we have the low, medium, and high dose. We have a very clear dose rate response. This means that the highest dose is protecting the crop better than the medium and the lower dose. So it's a very clear results, very good results. While the Zetanam product, that is the copper base, is achieving the best performance numerically, but not statistically. As you can see, the G letter is present also here in the highest dose of the LP plus the adjuvant treatment. While in this same trial, we have also the results about the bunches. As well, we have a very high value for this untreated inoculated. And this kind of trend, very clear about the dose rate response, is confirmed also in this case on the bunches. And the Zeteran product is confirmed as the best treatment. Passing to the Spanish trials, the contractor here is called Innovagro. We have performed a grape downy mildew trial as well. And here is the ADPC analysis again on the leaves. And here you can see that for the LP plus the adjuvant, we have a very clear situation where the highest dose performs better than the lower one, while the CP seems to works better on the medium dose, while the highest dose is not achieving the results that we were expecting. But in any case, this kind of results of the medium dosage it's a very good, it was a quite good control of the disease. And again, in this case, the copper-based product is the best performance. While on the bunches, again, we have the same situation, very good trend, dose rate response for the LP plus the adjuvant, while for the CP, we have this kind of weird results where the medium dose, it's performing better than the highest one. Then in France, we cooperate with the Science Agro-Atlantique contractor next to Bordeaux, as we saw before. And again, the ADPC in the leaves, here the results are not very clear. As you can see, we don't have a very clear dose rate response, but what we can say is that the best results are here, represented by the CP plus the adjuvant at the highest dose rate, while the other are performing as a moderate level of control of the pathogen, while the Zeteran product is always the best one. Here we reported the Salerno trials that, as I said before, was the first moment that we started to study and screening a new kind of formulation with different co-formulants that were compliant with a bioproduct. And we just reported here some results. We tested the LP20 peptide because it was possible to use it as it was we had some leftover of this peptide, while the CP20 was devoted just to the efficacy trials. And here is reported the percentage of efficacy reached by every formulation. And the best one was this one. As you can see, we have more or less 70% of efficacy, the LP20 plus the adjuvant number 3. And here is very important to focus on this point that this is the efficacy performed by the LP20 solo, straight, and with the peptide, with this kind of adjuvant, it enhances a lot its performance. And the Zeteran product always here as the reference. Okay, so we start to see some potato trials that was performed, one in Italy by Agricoltura 2000 next to Bologna. Again, we can see the ADPC analysis on the leaves. Again, here we have a good dose rate response. So the best results are achieved by the LP and CP highest dosage, and the copper is always the best treatment of the trial. And for Innovagro, that is the Spanish contractor, we have, again, for the LP, a good dose rate response, while for, and also for as well, is confirmed for the CP plus the adjuvant. And here also the reference performing very well. In this slide, we wanted also to analyze the data in a different way. So we decided to consider the last assessment of each trial from Agricola 2000, Innovagro, and Science Agro Atlantic, and to put it together and to consider together in order to obtain a medium data that could identify the performance of our peptide. And so here are the first box plot. I think that you are familiar with the box plot representation. The first box is representing the severity, that means the disease pressure in the field, and we reach about the 22% of the disease development and pressure. That is a quite good, moderately level of disease pressure, while the other three boxes are representing the efficacy of our peptide. So the LP plus LP20 plus the adjuvant performs well and obtain more or less 60% of efficacy against Plasmodium viticola, while CP20 plus the adjuvant achieved the best performance compared to the other peptide, about 64% of efficacy. While the Zeteran product, as you can see, performs very, very well. It's a remarkable results, about 21 plus 3% of efficacy against Plasmodium viticola. Sorry, this was about the leaves situation, while for the bunches, we consider the two trials, Agricola 2000 and Innovagro, and here the situation is about 41% of severity on the bunches, and here the best results are about the LP20 plus adjuvant. We arrived at 77% of efficacy, while CP20 reached 69.6%. And Zeteran is always confirming its very good performance. For potato blight, we have some worst results. We put together the last assessment obtained by Agricola 2000 and Innovagro. And as you can see, disease pressure is quite high, about 41%. And the efficacy reached by LP20 plus the adjuvant is about 40%, and also CP20 is not so outstanding in terms of control of the disease, while the copper is performing about 70% of controlling the disease. So as conclusions, we can say that for the grapevine, both peptide provides very good efficacy. So we confirm the activity of these two peptides against Plasmodium viticola, are both numerically inferior to copper, but not always statistically, as we saw. And the delivery system including could improve also the LP performance, as the coformulant that we are studying right now could also enhance the performance of this peptide. While for potatoes, the peptide efficacy is always statistically inferior to copper, and the dose rate response was observed and confirmed in almost all trials. But we don't have enough information to state which one is the best. So finally, we don't have the final winner so far between the two candidates. We can say that for sure, for us, these peptides works very well against Plasmodium viticola, while against the Phytophthora infestan we have some more problems in terms of controlling the diseases. But for sure, we are very interested in this kind of peptide that works very well, and we wanted to go ahead with further investigation to better understand the potentiality of these two peptides. That's all.
Thank you, Maria. It's my first time that I see in detail all this data, and I'm really impressed by the improvement of the efficacy when you had what you call adjuvant number 3. That I understood is not that the one that you use for the real field trials, right?
Yeah.
Okay.
We are starting to see some synergistic effect between this type of co-formulant and the peptides, but we are just in the first step of this investigation. So,
okay. So, but formulation can help a lot at the end for making the peptide even more active, and that's a really great results. So, thank you for all the data that you have shown. Do you have comments from the audience? So, Sandrine?
yeah, please. Yeah. Could be. We don't know yet, but could be could be a point that we have to investigate more. But for sure, the formulation is more complete and could also help to have a better stability of the formulation. So yeah, we have to go deeper in detail. For the formulation matter, we are just at the first step of investigation.
Maybe at the microphone so that also people can hear.
Just to add some information, here we are under field conditions, and field conditions in Italy, as in France and the northwest of Spain this year, were very conducive to downy mildew, a lot of rain. And before the last assessment that she showed, for 3 nights, we had almost 20 millimeters of rain every night. So we presume we are not sure yet that also the persistence, the ability of the peptides to stick to the leaves, because peptides are very much hydrophilic. So the rain fastness is an issue. The distribution on the leaves is an issue. The persistence is another issue. So formulation for sure may help a lot. And according to, just to reply to your question, Paolo, the adjuvant number 1 was the same as as we used in all the trials, and you have roughly 20% improvement of efficacy with the adjuvant number 3.
Okay.
So actually, formulation can help a lot in this kind of for several reasons. We don't know yet which one, but. Yes, the oral lipophilic, yes. Just to improve the wax, you know, the wax affinity with the leaves and with the bunches mostly than the leaves. And this adjuvant helps a lot to stick to the wax of the leaves and the berries. And the stability, yeah. As you know, we tested also the stability, not under proteases as you do, but under storage condition, the usual storage condition that we are asked to perform for any agrochemical. And after 2 weeks in 54 degrees, under 54 degrees, we had the stability, both the straight product and in water product, so without proteases inside. The stability of both peptides was pretty good. We continue for 3 months. We don't have the data for 2 years, but after 2 weeks, 54 degrees, you may assume that you have 2 years of stability. Usually for chemical for synthetic chemical products. We don't know yet if it works also for peptides, but this was pretty good, pretty tough test and pretty good results.
Thank you, Pietro, for your comments. Do we have other questions? Obviously, as coordinator of the project, to see that a company leader in the agrochemical sector is interested in our peptides is actually, it's like to say, okay, the project was good. At the end, we reached the objective that we wish to to have. Thank you, Maria. Thanks a lot. Okay, as I was saying, to have our industrial partner interest in our technology is somehow like to say, okay, peptides are really working and there are interest for also from the commercial point of view, the application point of view. So it was not just research in the lab, but it's a research that we can try to transfer really to the market. So as I was saying at the beginning of this meeting, we had 4 major aims in this project, in the NoPEST project. The first one was the identification of linear and cyclic peptides. And indeed, I think we reached this aim, as Maria was showing. She was mentioning LP and CP. LP means linear peptide, CP means circular peptides or cyclic peptides. And indeed, she has just shown us that those peptides works under real field condition. So far, we have tested just simply solving them in water. And it's really nice to hear that if you try to formulate them, you have even a much better performance. Sandrine has shown us that we can even improve the efficacy of these peptides towards peptidomimetics, so changing just 1 or 2 amino acids, making them much more stable and having a higher affinity for the target. Hanok has also shown that we have in our hands some non-peptide small molecule that so far we have tested under lab condition. For time reason, for budget reason, we didn't manage to go to the field, but this is something that we can keep going with those molecules and also transfer them to SIP-camoxone eventually in the near future and ask them to test them under field condition. And we have seen that all this technology that still it's rather costly at the moment, we can reduce the cost by combining them with tools for the precision agriculture. So sensor that will allow us to early detect the infection and probably to avoid to spray all these molecules in the entire field, but just on specific spots of the field. So essentially, in 2019, we started with a simple idea, and I have to say a simple idea coming from the mind of a very clever person that is Professor Simona Masiero. And we ended up in 6 years to a prototype that is now validated under field conditions. So based on the scale of technology readiness level, we move from 0 to 5, and those peptides are actually been patented. They have been, we submitted a patent in November 2023, and now we are at the stage of a PCT application that was just submitted a few weeks ago. And so it was 6 years, but I think were well invested, I would say. But there are still some limitation. As Sandrine was saying, at the moment we have around 18 peptides that are used for agriculture on the market. Actually, most of them are insecticides. They are not against fungi or mycetes. And the reason of that is that the technology we had so far require a lot of time to identify those peptides. And we mentioned several times during the talks their production costs are still too high to be applied in agriculture. You might need even more than 500 euro per hectare for treatment, and that's certainly not competitive and not economically sustainable for farmers. We do believe that with the NoPEST technology, we have addressed the first problem. You have seen by the talk of Simona Masiero that we can get a lot of candidates in really few months. Our technology, the technology developed in NoPEST, can move from an in silico analysis to the first candidates to be validated under laboratory condition in 6-8 months. And then obviously, as Maria has just shown now, we can transfer them easily to the field and test to the field. This is actually one of the images that we have taken during our visit to the field trial, the Sipcamoxon run in Piedmont region. And these are the non-treated benches, and these are the treated benches with our peptide. It was really impressive to see their performance under real field conditions. So concerning the speed to identify the peptides, I think we have solved the problem using this pipeline that we have developed in these 6 years. Really, in few months, we can have candidates, and then these candidates can be tested under field condition. There is instead much more work that needs to be done concerning the reduction of cost of production. This is actually the strategy that we aim to adopt in the coming years, because obviously we would like to continue with this technology. Of course, we need to search for fundings. But the idea here is to try to reduce the cost of production of those peptides, not using chemical synthesis like we have done so far. There are still, the costs are still too high, but allow microorganisms to produce those peptides. We simply clone the DNA sequence of this peptide into vectors, transform either bacterial or yeast cells, and put them into bioreactors and let them to produce the peptides. So we estimated that with this strategy, we could have a very drastic reduction in the cost of peptides, so that the cost will become competitive with respect to the traditional, or to the conventional pesticides. And so in the next months, in the next year, what we have to focus is obviously to optimize microbial strain, biomanufacturing protocol to produce those peptides into bioreactors. And when I say optimize microbial strain, I'm thinking also to optimize strain that can incorporate those modified amino acids that Sandrine was reporting. So also to be able to produce peptidomimetics using microbial strain. We just learned that formulation has a major impact in the activity of those peptides, and that's a sector where we need to also to spend time. And then, of course, also all the other classical assay and analysis that you must do before you can move to the market. Of course, we have some preliminary data on toxicological and ecotoxicological studies that we have run in the lab, but that also Sipcamoxon has run. We know, for instance, that those peptides are not dangerous for bees, and that's a very important aspect if you want to use in the field. And of course, we need to go with these that we can call recombinant peptides, test them again under field condition, and see how they perform and what would be the cost to test them under real field condition, with the final aim, obviously, to reach the stage of a commercial product. But I think the data and the results that we got are really encouraging, and we'll certainly continue on the way to improve this technology and to bring it to the market. Now, before concluding, I would like, first of all, to thank all the researchers that in 6 years had a major role in all the experimental activities in NoPEST. I think we have been quite lucky. We had also a lot of very young, talented research PhD students or postdocs that they gave a real important contribution to the project. As you might expect, as a relatively old scientist and professor, we don't have much time to spend in the laboratory. And so most of the research has been run by these young students, and they've been really working hard and quite effective, as we have seen from the results that we have got. And I'm really grateful to these young, talented scientists for all the work they have done. But I have also to admit that if you want to that a research project work as it should and respect all the deadlines, you need a very good project manager. That was actually, you might remember, we were in Brussels at the end of the first year, was our first review meeting, and the review panel was saying to me, look, the science is excellent, but you know, you need to organize a little bit better the project. So probably you need a project manager. And then it was the time we said, okay, we try to get a project manager. And I'm really grateful to Dr. Chiara Mizzotti that really took the management of the project, and she was really great in pushing us, in respecting all the dates, in organizing everything, including this final meeting. And obviously, before concluding, I really wish to thank, if I can, the Accademia dei Georgofili for guesting us in this wonderful place, in this very prestigious academia. And of course, I would like to thank Professor Massimo Vincenzini, so the president of the academia, for giving us the possibility to have this meeting here. Professor Amedeo Alpi and Professor Piero Cravedi that help us a lot in the organization. And so thanks to them, if today we are actually here at this meeting, and obviously Dr. Simone Milani that help us with all the small technical problem linked to the organization of these meetings. Thanks a lot to all of you. I think we have done a great job. I think we deserve applause.